Hello and welcome to class number 32 of the quick revision series for Prelims 2024. I hope all of you have attended all these classes and more importantly, not more importantly, but yes, also importantly, you have subscribed to this channel and also hit the like button. We are back again with another class of Science and Tech to discuss the current affairs of the last 1 year or so. Without any further delay, let's begin with the first one. The first news is a new test that has come up, very helpful for the TB patients. It will provide results in a much shorter period of time, lesser costly, and also it will tell us whether a certain patient is resistant to the first line of medicines. Now, as you know, tuberculosis is in itself is a very bad disease, but it has been enhanced even more, accentuated even more because of the fact that a lot of patients are now resistant to the medicines that are used to treat tuberculosis. For example, the rifampicin medicine that is used to treat tuberculosis, a lot of patients have developed resistance against it. Now you have to conduct a test first to see if your body is actually accepting that medicine or not. That test also costs a lot of money, it requires a lot more time. So there's an alternative that has been found to that. This new test is called Expert MTBRIF test. It will be a molecular test that will cost 20% lesser and will return the samples or will return with the result in a much shorter period of time. Interestingly, this is not the only test. There are other tests as well. MTB-XDR that is still used for diagnosing the most severe form of TB that will still be costly at about $15. Now, this particular test is expected to revolutionize tuberculosis, its diagnosis. Again, it will not stop tuberculosis, but it will at least be able to diagnose it in a much faster manner. It will contribute to rapid diagnostic. Thus, it will detect Mycobacterium tuberculosis complex called MTB. Along with that, simultaneously, it will see if you have resistance to rifampin, which is usually a drug given to treat tuberculosis. The result will take less than 2 hours. Standard culture usually used to take 2 to 6 weeks to grow, and conventional drug resistant tests added 3 more weeks. So usually it used to take more than a month to come up with these 2 separate tests. This particular test will do all these things together, both of them together, and will give the results within 2 hours. So it is expected to revolutionize diagnosis in tuberculosis. Second important news, messenger RNA or mRNA. Very important, has been in the news in fact ever since the COVID-19 pandemic began. When the COVID-19 pandemic began and the companies were trying very hard to come up with vaccine as quickly as possible, that is where new techniques such as mRNA came into the news. And now deservingly, the people behind this particular technique have been awarded with the Nobel Prize for their mRNA vaccine that helped prevent COVID-19 cases in a lot of people across the entire world. Now, what is mRNA? It is a molecule in genetics and serves as an intermediary between DNA and protein synthesis. How? So basically from the DNA it is taken up. It's a single stranded molecule. It only has nucleotides which is synthesized through transcription. The purpose of this molecule is simple: to carry genetic information from DNA up to the ribosomes. That is the entire job of this messenger RNA. That is how it works. The DNA, which is double stranded, from this RNA and mRNA is taken. It is translated, mRNA then translated into amino acids, which then results into ribosome. Amino acids form polypeptide chains and then they fold into different types of proteins. These vaccines have been regarded as much more effective as compared to other forms of vaccine. See, usually the usual method of building the vaccines was that you try and basically have or isolate those germs that are causing you this disease, those viruses. Try and inject those viruses in a very weak or dead form into a human body to help the human body develop antibodies against it. But mRNA vaccine does not do that. It basically prepares your own body to target those cells to target those viruses which are actually trying to harm you. They introduce a piece of mRNA corresponding to a viral protein. It allows the cell then to reproduce the viral protein itself, multiply, and it will help the immune system recognize the foreign protein which produces antibodies, and then it basically fights against the pathogen. These antibodies protect the body against the infection, they recognize viruses. So the chances of someone falling ill because of a weak virus being injected through a vaccine are eliminated here. Because as you know, for example, in vaccines such as polio, when you inject the polio virus in a very weak or even dead form, because of extremely low immune system in some kids, they might even fall ill because of that vaccine. In this case, in case of mRNA vaccine, this does not happen. Next, something else in news was danger analysis. Danger stands for deleterious and anticipatable guides evaluated by RNA sequencing. What exactly is this? Before you basically get confused, you all would have read a few years ago, there was a new revolutionary technique that came up called gene editing. CRISPR, gene editing, there were a lot in the news. What was gene editing? The simple idea of gene editing was that there are certain diseases that are passed from generation to generations because it is in the genes. So when you have certain diseases, your doctor will ask you, did your parent also have that? Because a lot of these diseases, they carry on from one generation to other in the genes. So how do you prevent them? So a new technique was found out that in order to prevent these diseases from spreading from one generation to the other generation, why not edit the gene? Why not make changes in the gene so that it does not get passed on to the next generation? This was done using the CRISPR technique. Now on paper it was wonderful and amazing technique, but to implement it, it had a lot of dangers. Implementing these kind of techniques at such a low scale or at such a minuscule scale is not very easy. So now a new technique has been found out to do the same thing. So it will do the same thing, it will just do the gene editing only, but it will do with a much safer approach. And that safer approach is called danger. It's ironical that the approach is safer, but the name is danger, but that is how basically it is. It's a new software tool that makes genome editing much safer by overcoming the limitations in the present CRISPR technology. This one, the new technology, works by conducting risk-free or risk-averse off-target assessment using the RNA sequencing data. It's a strategy that uses RNA sequencing, evaluates the effectiveness, potential of the target, and then it basically undertakes a gene editing process. The guide RNAs that are used in this particular process are those RNA sequences that guide to a specific target where exactly the editing has to happen. So something like this is how gene editing works. As you can see, the gene specifically designed synthetic part of the gene is taken away, an enzyme cuts off the target DNA strand, and a healthy DNA strand is basically then put in. So this is how basically it works. Again, these are technologies that are obviously much easier to draw on paper, much easier to theorize, but to implement that, to find tools for that is a much more complex process. It can put a lot of lives in danger. That is why it is good to have technologies which do it in a much more safer manner. So that is a DNA editing technique. Genome editing, also called as a gene editing, as I said, is a technology that allows a scientist to change the organism's DNA. Mainly the idea is to make changes in the gene, because a lot of diseases get passed on from gene to gene. It can potentially decrease or eliminate serious genetic diseases and reduce chances of generations being sick with the same disease time and time again. There are multiple techniques that are used. The most famous one is the CRISPR-Cas9 technique. Then there are TALENs, there is ZFNs, HDR, and now the danger technique has also come up. But these are the different types of techniques that are used. Again, the idea is the same, that you are editing the gene. How you are doing it, the technology that you are using, that changes from time to time, from place to place. But the idea of genome editing remains the same. Next thing in the news, Calyx coca. What exactly is this? It's a new vaccine developed for cocaine called Calyx coca. The idea is simple. When someone consumes cocaine, it goes into the bloodstream of the person. Now, it impacts the bloodstream also, but the major impact of cocaine happens when it reaches your brain. That is when the neurological system of the person starts to get hurt. So, the idea of this vaccine, Calyx coca vaccine, is that it will prevent flow of cocaine into the brain. So, it will not prevent it from going into the bloodstream, that will happen, but will prevent that from going into the brain. That is the idea. It has been developed by engineers and scientists in Brazil. The idea is to kill addiction to other substances and cocaine specifically. It generates antibodies that will bind to cocaine molecules in the bloodstream, and it will obstruct the cocaine from reaching the brain. This binding process, how will it stop it from reaching the brain? It will basically increase the size of cocaine molecules, and then it will be too large to enter the brain system. So it's a simple idea. Why not increase the size of the cocaine particles so much that it's just not small enough to enter the brain? That's how it will work. This area of the brain is typically stimulated by cocaine to release dopamine, and that is why it gives pleasure. That is why people get addicted to these drugs, because a part of the brain likes the feeling and it releases certain hormones that basically gives you a kind of a light feeling or a happy feeling, and you want to repeat that feeling time and time again. Individuals who receive the vaccine will not experience that effect of cocaine, and thus they will not take it again. That is a hope, that after you get this vaccine, you will not get that kind of a high or that kind of a happiness that you used to get with cocaine. So maybe, just maybe, you will stop taking cocaine because it is not giving you the kind of desired impact. Vaccine is moving in the final phase of trials. It has not been rolled out so far, but it might be rolled out soon after some trials being done on humans. Next news, thalidomide tragedy. What exactly is this? Australian government has issued an official apology to its own citizens for a scandal that happened many decades ago. So 1950s and 60s, there was a drug easily available in Australia. It's sold even today. That was called thalidomide. Now, why was it used? It was used mainly by pregnant ladies when the pregnant ladies experience nausea, etc. So, symptoms such as nausea are very common during pregnancy. In order to avoid that, many ladies do take medicines. One of the medicines available for that was thalidomide. It was much later found out that thalidomide actually had resulted in a lot of complications, including absent limbs in the baby, including blindness, deafness, and even malformation of the internal organs. So a lot of defects in the babies were directly related to this particular medicine that was being consumed in large number by the pregnant ladies. So that became a big scandal, that how come the government, even after knowing this, it is the government's responsibility to undertake trials before any medicine is rolled out in the market. So how did the government not stop it? How was it available for a couple of decades for this purpose? That is why it became a very big controversy. See, the same medicine that might not give any side effect to a male can have a huge side effect on a women, especially pregnant women. That is why pregnant women can't just have any medicine that she used to have before pregnancy. So it's not that this medicine itself is banned for everyone. In fact, this medicine is still available today because it has shown some results in treating certain diseases such as leprosy and different types of blood cancer as well. But it was just not suitable and safe enough for pregnant ladies. That was the entire controversy, and that is why the Australian government has come up with a sincere apology to its people. Next news, something called the phage therapy. What exactly is phage therapy that India might also start? Phage are kind of viruses that are used to kill bacteria. So very interesting. Phage are kind of viruses that are used to kill bacteria. Now we are living in an era where antimicrobial resistance has become a very big problem. We consume so many medicines and we are still not able to treat the diseases because our bodies have gotten so used to it that we have developed these antimicrobial resistance. That is why phage therapy is being seen as a ray of hope for those specifically who are suffering from problems such as antimicrobial resistance, because what you will do is, rather than using some medicines, you will use some specific viruses that will kill bacteria in your body, called phage therapy. So again, as I said, simply defined, it's a medical treatment that uses viruses to target and destroy harmful bacteria. The other part is, one of the other problems that we have with antibiotics in our body is, when you consume antibiotics, it kills almost all types of bacteria. Now the problem is, there are some bacteria which are not harmful for your body. There are some bacteria on the contrary which are actually helpful for your body. But antibiotics don't differentiate between good and bad bacteria. They kill all the bacteria. That also becomes a big problem. So this phage therapy will infect and kill specific bacteria only and will leave alone the beneficial ones and will ensure that people can be treated even if they have antimicrobial resistance. This is still a new kind of a therapy. It is being implemented in some very small scales, but not in India. We have to see how it is adopted in India in the coming years. Usually, you do not see India being any first country to adopt new technology such as in treatments of patients. We usually wait for the other countries to adopt, see the effect of it, and then we take a decision. That is why we can see it in India as well in the future. It is also called as bacteriophages because again, they are designed specifically to replicate and to kill these bacteria cells. Phage therapy is not very new. It goes back to 1917 because again, it can identify those bacteria that are harmful for our human body and they can actually help us to destroy them and have a much healthier life. Next, the Government of India, the National Health Authority, has launched 100 microsites project across the country. Now, what is 100 microsites project? The aim is to accelerate digital health adoption, to make sure people use much more technology and try and help the healthcare system also to reduce some of the burden. Launched by this National Health Authority, the idea is to boost digital health adoption by establishing small ecosystem with complete ABDM integration. Here ABDM is Ayushman Bharat Digital Mission. So again, ABDM is Ayushman Bharat Digital Mission. What are microsites? Which we are talking about, 100 microsites project. So it's a cluster of interconnected healthcare stakeholders where healthcare providers like hospitals, clinics, doctors will be connected to labs, will be connected to pharmacies, etc. within a defined geographical area. So within a, let's say, a city, all these will be connected online. All these, for example, can share reports with each other. Let me give you a simple example. Let's say that you go and undertake a diagnostic test, okay? You undertake a blood test or an X-ray, something like that. Now you have to take the report and then give it to the doctor. You might not be able to see the doctor might be busy. What happens in this case when all of this will be connected? The doctors will be able to see your reports online from that lab itself without you having to do anything in between. So when all these are connected, the pharmacy, for example, would automatically get to know that, okay, this doctor has prescribed this medicine for this patient, rather than you having to then take your prescription and say, this is the medicine I want. So that is how interconnectedness would work. That is the idea of a microsite. So the idea is to develop 100 such microsites across the entire country. It addresses adoption challenges. It encourages private sector participation as well. And it again is dependent on whether all these parties are interested, how they are using technology, how they are coming up together. The idea is to get technology adopted at a much basic scale or at a much more ground level, reducing the need for any human intervention as such. The previous implementations were done in cities such as Mumbai, Ahmedabad and Surat, and now the idea is to have this platform across the country. See, these kind of things, in order to work, you have to first take all the stakeholders together. Everyone has to agree that yes, we want to be a part of it. Then you have to basically get them on a same platform, on the same system, make sure all of them can use technology, train them, provide them any hardware, software that is required. Because see, you can't expect any of the stakeholders to spend extra money, because they're not going to earn extra money from this. They are not basically getting any profit out of it. So why will they invest money? So it is the government that has to make sure they are funding it enough. The government has to make sure that they are making it easier to be adopted. It will help in improving transparency, efficiency, data access, and again evidence-based decision making. The doctors can experience it. The doctors can see the reports quicker. You don't have to go to medical stores and then basically the medical store will only give you the medicine that is prescribed by a doctor. All this will happen under this project. Next news, India's AI supercomputer called Eravat has been added to the list of 100 most powerful supercomputers in the world, which is a big achievement. Eravat has been ranked at number 75 in the top 500 list. The list is developed, or the list is produced by International Supercomputing Conference in Germany. Eravat stands for AI Research, Analytics and Knowledge Assimilation. It's an AI-based cloud computing infrastructure which has been installed at CDAC in Pune. This is where most of India's supercomputers are kept. That is in CDAC Pune. It is running on the Ubuntu operating system. Just like you have the Windows operating system usually, or you have the Android, etc. There's an operating system called Ubuntu. It has a potential to use AI at a much larger scale. Usually when the governments make supercomputers, one of the biggest applications are always weather forecasting. Because in order to forecast weather, you need to process a lot of data. Humongous amounts of data has to be processed in a short amount of time. That is where supercomputers come in handy. It's also expected to help in drug discovery, artificial intelligence research, anything that requires calculation of humongous amounts of data, big data as it is called, and then come out with results in a specific period of time. That is where supercomputers find their application. The top 500 list is a common list that talks about 500 fastest computer systems in the world. It was started in 1993, has been updated every 6 months after that. Please do remember, it's based on a benchmark called LINPACK benchmark. LINPACK benchmark basically measures a computer's floating point rate of execution, means at what speed can it execute the instructions given to it. The smaller the speed, or the smaller the time taken rather, the more will be the speed. India has other famous supercomputers as well. We have something called Param Siddhi AI supercomputer, again at CDAC Pune. It was ranked at 131. We also have Pratyush supercomputer ranked at 169. It is at Indian Institute of Tropical Meteorology. We also have the Mihir supercomputer, which is at 316. It is at the National Center for Medium Range Weather Forecasting. As you can see, most of it has been used, or the application is mainly for weather forecasting purposes, because that is again something that requires a lot of data processing and a lot of people's lives depend upon that. Next, number 339, a new issue of blue bugging has started. Andhra Pradesh government has cautioned the smartphone users of blue bugging. What exactly is this? As the name suggests, related to Bluetooth. So basically, when your Bluetooth device is discoverable, means anyone within the 10-meter range can actually see that your Bluetooth device is accessible or it's available. They can try and pair with it. Once they pair their device with your Bluetooth, what will happen? They will gain full access to your laptop or to your mobile. They will be able to send and read messages. They can access your phone book. They can even listen to your calls. All of this just by Bluetooth pairing. This is called the blue bugging. Earlier it used to impact laptops, etc., but now it has been impacting mobile phones as well. Now the good part for you, for us, is that it's a technology that can only be used in a short range, because Bluetooth only has a range of 10 meters. So anyone that is trying to control our device has to be within this 10 meters. So you can easily identify who is doing this if something wrong is happening. Someone sitting far away cannot do that. That's a good part about this. There are some other related problems related to Bluetooth. For example, this blue jacking. When this is a simple pranking technique where the device owners are pranked by sending harmless messages. There's blue snarfing, or the hackers access the data without your permission. It will download sensitive data from your mobile or your laptop. There is juice jacking. It has also become a big problem now. You would have seen many people tell you should not use USB devices to charge your phone. When you don't have a charger, what do? You basically charge your phone by putting a USB device and then connecting it to laptop. Laptop acts as a power source, right? But when you connect, let's say, your phone to that laptop then would have the ability to take data from your phone. This is called juice jacking. When sensitive data is being taken or a harmful software is installed in your phone for later on purposes via the USB charging port. This is called juice jacking. Either data is taken away from your phone or either some virus or some malware is sent to your phone. That becomes problematic. All these are different types of issues that are seen as being used by hackers in today's time. Last news for today is NAFIS, National Automated Fingerprint Identification System. This is used by NCRB, National Crime Record Bureau. This team of NAFIS has recently won the Gold Award by the Government of India for implementing the technology in the best and effective manner possible. What exactly is this system? So it's a system that was launched by the government with the purpose of creating a database of all the criminals in the country with their fingerprint. Just like we have our DNA, which is unique to us, similarly fingerprints are also unique to individuals. So the idea is anyone who has become a criminal against whom FIR has been filed, you take their fingerprints in the police station, you identify them by giving them a unique 10-digit number called 10-digit national fingerprint number to each arrested person for a crime. It can be searched in a database so that tomorrow if someone has been arrested, you can basically check their fingerprints by this unique number and see if they were arrested earlier as well. Throughout the life, a person's unique 10-digit number will remain the same. It will have palm print, it will have fingerprint enrollment, portrait capture. All this will be a part of this data. It's managed by National Crime Record Bureau at the Central Fingerprint Bureau that is in New Delhi. It is being done, as I said, to have a proper database of criminals who have been arrested sometime or the other to ensure that they cannot forge their identity. NAFIS within 24 hours will give 10-digit unique number. So it's not instant, it will take about 24 hours, but it will give you a unique 10-digit number to everyone who has been arrested and who has been taken into custody. The unique ID will remain the same for the lifetime of the offender. Whatever crime that you commit will all be registered under different FIRs but to the same unique ID, the national fingerprint number as it is called. So whatever crime you commit doesn't matter. It's kind of an Aadhaar number for criminals specifically. The first 2 digits of the ID will be the state code of the state where the criminal is registered, and then followed by a sequence number which will be different for different people. So first 2 digits will be the same for a lot of people depending upon which state the crime was committed. The later on numbers will then be different. This brings to the end of today's class. I hope you learned something new today. We have a few more classes coming up before we wrap this up. I hope this has been helpful for you. Do subscribe and do become part of the Telegram channel to get these PDFs for your revision. Thank you so much. Bye-bye. Jai Hind.